Hi, my name is Yan Ko. I'm a PhD student at Dr. Avi Mayan's lab at Icahn School of Medicine of Mount Sinai. In the previous lecture, I've introduced the pipeline of top hat and cufflinks to analyze RNA-seq data. Once we construct the transcriptome, we will be able to apply various statistical methods towards a deeper understanding of your data. In this lecture, I will introduce the CummeRbund R package that comes with the pipeline for the downstream analysis. To remind you, here's a pipeline of top hat and cufflinks. We have learned to align the RNA-seq short reads to the reference genome using top hat, example the transcriptome, and identify differentially expressed genes using cufflinks and cuffdiff. So the CummeRbund package was developed by the same research group. It directly takes the output from cufflinks and provides functionality to visualize the transcriptome data and several frequently used data analysis approaches. Here's a list of points we will cover in this lecture. Since CummeRbund is an R package, we will first need to learn some basic operation in the R statistical programming environment. Once we get familiar with the R language, we will load the CummeRbund package and learn how to evaluate the quality of the RNA-seq data. We will also perform the hierarchical clustering as an example of downstream analysis. And also to remind you, here is the data we have. We have knockout and wild type at three different time points, G1, G2, and G3. So, what is R? It's a language and environment for statistical computing and data visualization. It provides a wide variety of statistical and graphic techniques. It produces publication quality plots and is easy to manipulate. There are many well-developed packages. It produces publication quality plots and is easy to manipulate. R is an open resource project, which means that you're free to develop your own libraries or packages. And you can share it with everyone else. There are many well-developed packages for many kinds of statistical analysis methods and free to use. You can also obtain the source code and rewrite it to suit your own tasks. However, compared with the well-maintained commercial software such as MATLAB and Mathematica, there is no guarantee of the quality of an R package. As a beginner, I suggest to use packages that have been frequently used and widely tested, or write your own function for better control. Fortunately for biologists, we have the bioconductor, which collects R packages performing high-throughput sequencing genomic data analysis with almost all well-established approaches. Several packages will be needed for CummeRbund to be fully functional. Similar as other programming language, we can simply write R code in a text editor and execute with R. However, this is not the best way to analyze your data with R. There are several graphic user interfaces become available in recent years, and I highly recommend the R Studio. We will first look at the R Studio workspace.
This is the interface of R Studio. On the left, we have the console that we can type in any R command. On the right, you have workspace, history on the top, and files, plots, packages, and help on the bottom. I will talk about these tabs in a minute. Let's first create a variable and assign a value to this variable. I want to give a value of 5. Hit enter. Nothing shows up here, but if we look at the workspace, we can see A shows up here, or we can also type A, hit enter in the console, and the value will show up in the console. Now let's create another variable B, give it a value of 3. And we can also do a simple addition of A and B and give the value to C. Now C equals 8, which is A and B. And on the workspace, we can see all of them listed here. We can also import data in this workspace, for example, from a text file in your file system. All the other buttons, we can delete the variables or save the workspace or open a saved workspace data. Now, if you have a bunch of code suppose you don't want to execute them line by line in the console, you can create a script and type it here. We can also execute the lines. Just move your cursor to the line if you want to run and click Ctrl plus Enter. It will go to the console and execute in the console. And now if we look at the workspace, the value of A changed to 6. This History tab records all the previous commands we've executed, and the good thing about this History tab is that we can click a previous command and click this, for example, the C equals A and B, and click this To Console button. And this command will go to console, and we can execute it again in the console by hitting Enter. We can also select a series of commands and click to source, and all these commands will be pasted to the source, to the script. This is very handy if you want to repeat some previous analysis. We can also open a history file from an existing file. For example, if you're working on another project, and you can import all the previous commands you've executed for that project and integrate to your current one. Now move down to the Files tab. So the Files list all the directories in your computer. This is the same as you were browsing within your system. For example, you can move around, go to the subdirectories. The good thing about this Files tab is that, so by default, R needs to know what's the working directory. And then when you refer to some data files or history files or scripts, R will look for these files under this working directory. If you want to set the working directory from the console, you need to type in the set working directory function followed by the path to wherever the folder is your data stored. Now, if you're using this Files tab, we can first move to the directory where you want to work from, and click this More button and look for this Set as Working Directory. And now, once you click it, RStudio will set up this folder you can see here as the working directory. It saves you the trouble of typing this long path names. The Plots tab will show any plots you did any plots. We will give an example later. And this package shows all available packages that you can load from RStudio. Most of them comes with R when you install it, but if you have some customized packages, it'll also show up here. You can easily upload a package by click the checkbox here, and it's equal to typing this library function, and you can check it again. Now this package will be detached. This is handy to load packages instead of typing in the console or in the script. Now the last tab is the Help. It shows the help documentation of the functions. You can search the function from this search here, and you can select from a drop-down list. Now I want to show you the example of plot. So let's search for plot. Now the good thing about help function is that it gives an example of how to use this function, and you can just simply copy and paste the entire thing in the examples and execute in the console. Hit Enter, and it will generate the plot. This is the basic 2D plot. You can zoom or export as an image or a PDF. Now if you have a fancy plot and you can export as a PDF and edit in Illustrator or Photoshop, and it gives you publication quality figures. Now we've seen examples of create variables and a basic plot. R, as a statistical programming language, R also offers the basic math operations, for example, log and exponential. You just type in the function as you do with the math. Now, except for a single value, now I want to create a series of values and want to assign these 5 values to the x variable. I can use the C stands for concatenate. I give it 5 values. Now let's see what's the value of x. It has 5 values in the array. And you can also type in the console. It's the same, 5 values. Now, in the workspace, it shows it's numeric. You can also check the type of values using the mode function. It says numeric. R also allows us to take the keyboard input from the using the scan function. For example, I type scan and it gives me the index, and I can type. I leave the last one empty, and now Y has 6 items. What if I want to create a series of data that follows some rules? You can use the ChIP function. For example, I want to start from -1, goes up to 1, step by 0.1. Now we have exactly what we want. Well, the ChIP function can also give you more complicated rules. For example, I want to have 40 elements starting from -1 and step by 0.5. Now let's look at Z1. It has exactly what we wanted. Another useful function is the wrap function, that we can create repetitive patterns in the array. For example, if I want to have an array has 10 3s, I will type 3, 10, and now Z2 has 3 by 10 times. We can also, except for numbers, we can also create a character array by using the wrap function. Let's give Z2 another value. Now we have 10 times of A instead of 10 times of 3. We can also give a fancy rule to the wrap function. For example, I want to have twice of value from 1 to 6. This column stands for 1 to 6. And now we have 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. We can also put the wrap function inside a wrap function to create an even more complicated array. For example, here I will have 3 times of each value and comes from 1 to 6. So we've seen that R has the numeric and the character type of variables. We can also have the logic variables in R. For example, here I give A a true value, and when I check the mode of A, it shows logical. So basically we have true and false for the logical variables. And then we can also create a character array. So now we have A, B, C in the C variable, and the mode of the C variable is character. But when we're dealing with biological data, we'd like to see, like in the Microsoft Excel file, we have a table contains the data and the headers of the columns or the rows. indicating what is the data. In R, it's very easy to manipulate the headers of columns. For example, let's first create a variable called birthday and give it some numerical values, that's the year, month, and day. So now I want to give this 3 columns a header for each. We use this named function. So the name of birthday is now given by an array of characters, which are year, month, and day. Now if we check the names of birthday, which is the headers of birthday, we will see year, month, and day. We can also retrieve the column's value by using the headers. For example, if I want to see the month of the birthday, I can retrieve by using this month header and it show me it's 5. Next, I want to show you how to import your own data, do a basic plot, and export the variables from R to a text file. So first, let's look at the example data. So we have 2 columns of values. The first column is X values, the second column is Y values. So we're going to plot a scatter plot of the data point. First, we need to import the data. We will assign the data to a variable called data, and we'll use the read table function. First, give it a name of the file, and keep the header from the file. Here, T stands for true, and the data are separated by tab. This /T stands for tab. Click enter, and you can see that data is already here in the workspace. And we can click on it. It'll give us an overview on the left. And if you notice that this click also equals to the view data function. We can also, if you have a huge table, we can also see the first few lines of the data by using the head function. And we can also retrieve the column by using the $sign followed by the name of the variable. Now let's plot. Here I just want to do the basic plot. So I give the X values and Y values in the correct order, and it will show up under the plot tab. Notice that the labels for X and Y axis is the default, and I want to change that. So let's do the plot again, and this time we specify the X labels and Y labels. We can also specify the color for the data point using the color parameters. Let's say dark red. And notice that instantly the X and Y labels and the color of the data points changed. If you don't like red, we can plot a blue data point. So the plot function also gives some other parameters that we can set up, except for the X and Y labels. Suppose we've made some changes of the data. Now we want to save the changes to another file. We'll use the write table function and first give which variable we want to save and what's the name of the output files, and we want to separate the data by tab, and we also want to keep the the quote, so set up the false for quote, otherwise R will give a quote mark wrapping each data. And I want to keep the headers, so I set true for column names and false for the row names because they don't really have the row names. And we can see it's already here. Let's take a look what's in the file. So this is exactly what we expected. Now, what if we set up the row name as true? What's going to happen to the output file? We can see that R automatically assigns an index for each of the rows. This is not what we want, so we set up as false. So instead of searching for the help documentation in the help tab, we can also use the question mark followed by the name of the function in the console. Now as we type in the help function, the help documentation will show up here on the right. So the last thing I want to show you is how to install packages. RStudio offers the handy way to get it from the user interface. Now let's type in the name of the packages. But usually it will generate a drop-down list, and we can select the package from the drop-down list and click install. It will automatically install the package. Yeah, so now it generates the drop-down list. But for example, if we type CummeRbund, it couldn't find automatically. So we have to go to the bioconductor to see how to install CummeRbund. Let's move to bioconductor. And search for CummeRbund. Select from the list. And here it shows you how to install the CummeRbund package. So basically we just need to copy paste these 2 lines of code, hit enter, and it will automatically install the packages. It will first download the packages and then install it. So here I already have the CummeRbund, so I'm going to skip this step